think next I'm coming up. And maybe just to follow on this comment from Ken, I think there will be a lot new challenges at the level 1 that we don't know about right now, especially with new molecular tests. For the first time, we'll get with a molecular test at the level 1, and I think we'll be smarter in 5 years to come again, but let's at least take what we have learned from the last 5 years. So now we'll switch gears a little bit. We had an excellent overview over what it takes to get to a comprehensive solutions. Let's take a step back and actually look at the technologies again. What do we need in terms of technologies? And I just wanted to briefly review the development process of the TPPs. Many of you are familiar with it, so I'll go through that quickly. Then look at the changing needs that affect the defined TPPs as they have been published, and then other novel needs as we perceive them today. And this is by far not a complete list, but I'm trying to get at it. So just quickly, like if we think back how the TPPs were defined, it was essentially that in, I think 2011 or 12, people sat together, global stakeholders, and made a list of what is necessary for TB diagnostics. Then that list that encompasses like 10, actually 9 different tests there, one was split out to a 10th. Were taken and were looked at. So they span essentially the entire cascade of the healthcare system, so from the community health worker to the reference center, with most of them being in the lower levels of care. And looking at those 10 different needs, essentially global stakeholders were asked about what should be the top priority. So what the Gates Foundation is trying to do right now internally was attempted here with really having researchers, national program officers, market experts, and implementation scientists together to find out what should be the topmost priority and what is also most feasible within the next 5 years, considering a development timeline of 5 years. And out of that came a list of like 3 TPPs. Again, one was split out to include drug susceptibility testing. And all of those were focused on the point of care. Then the next step was that from those defined tests, target product profiles were developed, essentially a laundry list of characteristics, minimal and optimal. And those, again, were run by a global stakeholder group to identify really the TPPs as they would have impact, likely impact in the settings of need. So where do we stand with that now? We know that those TPPs were published in 2014. Again, like a publication is here available on the desk as well. And the TPPs were envisioned to be a strategic planning tool, a communication tool, and also tracking tool for progress. And they were also envisioned to be revisited as there's progress in science happening. So they were considered to be a dynamic document. And so the question is now, has there been substantial change to influence the TPPs as we have defined them now about 2 years ago? Do we have to go back through this whole process, or can we amend them and look at them with different eyes now? So I would say that, if we look at like the biomarker tool and the triage tool, TPP, that probably we haven't really had substantial new learnings to come back and say, okay, let's revise these. But I think if it gets to the DST tool, I think we have to consider the changes in the drug landscape. And I remind you that the drug susceptibility test that was defined in the TPPs in terms of drug prioritization focused on rifampicin over fluoroquinolones, including moxifloxacin there, and INH and PZA kind of rated equally. That was at the time when we didn't have the REMOX trial results published. So now we know that the REMOX trial unfortunately was not successful as we were hoping it to be. And the question comes up, obviously, are fluoroquinolones still as relevant and should be as prioritized as they were at the time? And my argument here would be yes, but please challenge me on this, because they are still a critical part of the MDR regimen. They're a critical part of the shorter stream regimen, which definitely does require drug resistance testing in order not to result in quick development of resistance. And also the PAM-C, there the jury is still out, but we are hopeful that the PAM-C regimen will be a regimen that will be coming out in the next few years. We know that the trial is on hold, but there are certainly other regimens also that contain PZA that are quite hopeful. And I think obviously we have the one that exists, HRZE. The impact of PZA testing there is debated. STREAM certainly has PZA as an important component. The NC005 also has PZA in there. There's bedaquiline, protonamide, and PZA in there. And that is a promising regimen that's being evaluated in earlier trials. What about other drugs that we haven't had in that list on top here? Bedaquiline comes up over and over, and we are very excited about that novel development. It is part of the 2 regimens that I'm listing here. And then also the linezolid, it's part of the NIX regimen of the TB Alliance, and also used as in many XTR regimens. So should we be looking at those drugs to be included in rapid molecular DST going forward? I think the other, so I would say probably we should, but I think the other thing that we need to consider, and you, I think, heard a little bit about that yesterday in the context of the RESAC-TB review, and we'll certainly hear about it more over the next days. But what do we understand about the genotypic basis of resistance? And I just try to highlight the top drugs here. I think for RIF, we probably know more about the genotypic basis of resistance and the importance of individual mutations than we know about the phenotypic DST. And I would argue that probably molecular testing for the RPOB is the best single reference standard. For INH, it's a different ballgame. There, we know that if we target the key drugs, we get only so much sensitivity while a high specificity is possible, but we would have to target a lot more to really get to what we are able to do with phenotypic DST. Fluoroquinolones, difficult topic. There I think we have as much of a problem with the phenotypic DST as we have with the sequencing. Because particularly for moxifloxacin, we don't quite understand what the best clinical cut points are and what certain mutations mean in the context of clinical outcomes. And also we don't quite understand cross resistance. And the similar issues apply also to second-line injectables. PZA has been discussed for a long time. There also, I think the jury is still out, but given the poor performance of our phenotypic DST or enzymatic assays, also potentially reference, the sequencing might be the best single reference standard. Other drugs like the protonamide, bedaquiline, linezolid, I would say that there we have so far incomplete understanding of the basis of resistance. So we have to kind of look at our wish list with this understanding in mind and have to look at it critically. And as you have heard yesterday and also this morning, there's a lot of work trying to inform a better understanding of the genotypic basis of resistance. That is ongoing, but I think it will be ongoing for a number of years, and it will be a constantly evolving understanding, and we'll also have to think about that in respect to the diagnostic tests that we are looking at. So, oops. Sorry. Okay. Well, yeah. So, if we look at this now, telling you that yes, molecular tests or sequencing might actually be for some of the drugs the way to go, but for others, our understanding is incomplete. Can we consider sequencing actually as a diagnostic tool? You've heard about it already a little bit or a lot yesterday as well. It's certainly used for surveillance already, and you'll hear more from Chris Gilpin from the WHO about this work of the WHO tomorrow. But what about sequencing as a diagnostic tool? Certainly, right now it's a very complex technology and it has different components, and it would be, I think, wild thinking to assume that within a 5-year time span we can get sequencing into a box like Expert. But can we actually get to a modular solution? I think that appears realistic, where we simplify the different aspects and optimize them and essentially have a modular solution that targets the extraction and PCR, the sequencing, including the library prep. We've heard from Wave 80 that there's work on that ongoing already. And then the data interpretation is a very difficult aspect. There is a lot of work needed to standardize the output and to really get to an output that informs the clinician and not just says, yes, we have discovered this mutation. That is not what we can work with in countries where many providers are computer illiterate and will not understand what a mutation result will mean. So a lot of effort needs to be put into, and is already put into, data interpretation, into standardization of language. And then we need to consider like the cost of this, obviously, and there are trade-offs between cost and throughput, but all of this is currently being worked on to put into a TPP, and Jim, you gave me a nice segue into this. I won't go into the details of the TPP today, but we are working on that, find internally, and very close to actually sharing that with a larger group, and we hope to get input from the larger group of technology and healthcare systems expert in this room, so to get the TPP and sequencing to the next level. Centralized integrated DST, I think that's also an important point to touch upon. The TPPs that were published by the WHO are point of care, so meaning level 1, level 2 at most TPPs. Centralized integrated DST would be at the reference level, or maybe as low as the district level, but certainly not lower. And what would be expecting from these tests, and how does it differ from a point of care test? I think we would argue that at those levels there is individualized patient therapy possible, and not just regimen-based selection. So for individualized therapy, one would have to have more information on different drugs, and that's why you see the drugs tested here being a larger list. But again, with the caveat of the knowledge that we have underlying for these drugs. And I think an important part for these centralized tests is that they have polyvalency, so that they have the ability to detect other diseases such as HIV. And I think there are many countries out there that already have a centralized system, and they will benefit from a combination of point-of-care tests where those can only reach, and centralized tests where their infrastructure is well developed. But certainly for many countries out there also, the infrastructure and all of the issues that Madhu touched upon, with either getting the patient getting lost, or the sample getting lost, or the result getting lost, apply to those, and there probably point-of-care testing is more suitable. But I think centralized tests have a role, but I think we need to look at them in terms of in comparison to sequencing. What are the benefits and shortcomings? I think sequencing has one key aspect where it might trump the probe or array-based assays, which is its adaptability of the platform. And that will also be a decision, obviously, of the regulatory agencies, including the WHO, of how to handle new information integrated into a test and the revalidation necessary for that. And obviously, molecular diagnostics have the one key, I think, aspect in that they can essentially simplify more at this point, that they have the potential to get down to the microscopy level or district level. Where I think targeted sequencing or whole genome sequencing, I just picked targeted sequencing as an example here, has quite a long way to go. And I think thinking about centralized testing, we need to think about the necessary links. So optimized sample transport to ensure that the sample, the contamination is low, the recovery of the mycobacteria is optimized, and the workflows are optimized. There's a lot of work going on in this, and this is also something that the WHO is keen to review next year, and we have an ongoing trial that's just starting on improved sample transport solutions. And then definitely ICT, so optimized communication solutions, to aid in the various aspects of the ecosystem, the diagnostic ecosystem, in communication to the patient, to the provider, in communication of tests that are, or devices that are failing, or training needs, or in the aspects of surveillance. There's lots of opportunities that need to be addressed, and there's a lot of work going on in that as well. I have a couple of other slides. I think in the interest of time, given that we haven't touched upon LTBI and treatment monitoring yet, I will actually skip on these. But if you're interested in that, then I'm happy to talk about this with you individually. Lots of slides. All right, thank you very much. Any questions for me? If not, then Rob.
You haven't touched on the quality of the specimen. Because that's, we did a little bit of a review. They wanted to get a training manual in South Africa for getting samples towards the expert. And just looking and going through the NHLS laboratories and asking what are the problems. One of the major problems is filling in the request form correctly. Yeah. And another one is what's the specimen type and its quality. Yes. It's a major.
Yeah, no, thank you for that point. So I would say I've touched upon it a little bit with needs for the sample transport up here, the optimized sample transport, because yes, the quality of the specimen is a huge issue. It's a huge issue in South Africa in getting samples to the expert testing. It's a huge issue wherever you go, ship a sample. It's the delay in time that the sample is sitting around, the cold chain need. It's, as you say, also the documentation need around that, and all of those. And some aspects can be addressed, I think, through technology solutions, and that's what we are looking at with this solution. Some aspects are more systems issues. And I think like tracking of samples is an important aspect. So all early on, like scanning something at the site of collection to potentially track it from there, whether there are optimized solutions to get to that. I think we need to think a bit more innovatively, and also think of how the FedExes and DHLs of this world are doing this, which also ties into the like supply chain aspects. But I think it's a very important point. Yeah. Sure.
I'd like to add on to Rob's comment, because I think we have to go back to the actual patient sample. In the old days, when I was a TV tech, we used to take people and bang on their chest and turn them upside down and you know, and stand there and coach them, and that seems to have disappeared. And so it's not just once you got it, but getting a decent one. And with HIV patients and the smaller children, it's even more difficult. And I think better instructions and more specific paths to follow for the caregiver to get the best sample out of the patient, I think, is something also that could be developed. Some kind of little video for the patient as well. And then the second thing I want to say is that Stanford, many years ago, we did develop an HIV sequence database. Bob Shaker put it online. It's free to everybody. And you put your sequence that you got off of whatever system you used to get the sequence, and it will look at all the data on patients with the same mutations and their outcomes, etc., and actually deliver you in text an answer which says, your patient is best likely to be treated with these 3 drugs, and here's their likelihood of improving with regard to HIV. And I think that same thing could be done with, hopefully with RECE.
Yes, and thank you for making that comment. That's certainly a goal as well, to essentially optimize the platform to inform providers directly. Yeah. And the Stanford database for HIV is widely used, and certainly a good example. Martina.
Hi, Claudia. So I just wanted to you to clarify a little bit regarding the updates and revision of the rapid DST target product profile, because what I see from my hand is that compared to 2 years ago, the main changes that we have seen are, yes, the fact that the remarks trial was not as successful as we were hoping, and the possible bigger role of a shorter treatment regimen. So I don't see many changes that would justify big revision of this TPP. Yet I know that we were not as successful as we hoped to push out a clear message to the R&D and test developers community. I'm hearing people being still confused in terms of, I mean, the last was also the discussion with CFID last week about not having clear which kind of product and which kind of drugs they should include in their products. So I just wanted to have a little bit your opinion on whether there would be really a need to revise the TPP, and if this is the case, how we should go about it in terms of being sure we make a thorough assessment and we are sure we send out a clear message to the test developers so that we are not stuck in this back and forward situation.
Yeah, thank you, Martina. I see Chris hiding away there, just thinking of all of the work that has gone into these TPPs. So yes, I mean, the changes are probably not that substantial to require the whole process that we went through initially, but I think we should ensure that the discussion is ongoing and that the global stakeholders really have a say in this. And the CPTR here, the meeting, is a great forum to do that as well as the work groups. But I don't think at this point it's the time to go through the whole process again. But I totally agree with you that we need to be clearer in our message. And I mean, I would argue that the changes that I proposed are add-ons. They are not changes essentially. The drugs as they were identified are still like the top drugs, and I think also while on the one hand we are trying to align diagnostic test development with the treatment development, I mean we've also seen that unfortunately one regimen that we were very excited about has failed. And so it's a cautious weighing of like what do we have available right now, and for those we need these diagnostic tests, and what is likely going to come. And in that I think fluoroquinolones, PZA, especially PZA, are important components of novel regimens to come. So I don't think it has changed too substantially. And yeah, we need to be better in that messaging. All right, last question, because I know we are already 20 minutes late. Larry.
Claudia, could you say something more about your thinking about combining, at the time of initial diagnosis of TB and HIV? Because that seems really to be constantly left out of the conversation.
I mean, I think it's especially obviously in sub-Saharan Africa a critical component. I think we are thinking again way too much in silos there in terms of like TB, HIV, also the way how the healthcare system is set up. So I think it's absolutely crucial. And that's why I also mentioned, particularly for the centralized platforms, we need polyvalency and need to test for TB and HIV, and we can't have one box for this and one box for that. Ideally, it should be on one platform. And I think many test developers are moving towards that. David, very, very quick comment, please.
A very quick comment to that point. We are meeting at that point in competition between TB labs and HIV labs. So each of them is somehow worried about the budget. So that's really, I think, one of the hardest points to go through, that there is a consensus between a TB and an HIV lab to combine the diagnosis. At the moment, they would stand in competition. Who is taking over which part of diagnosis? So, well, when you're talking molecular, it would be quite natural that the HIV lab has more molecular background than a TB laboratory. But, well, will the TB laboratory give away the samples?
Yeah.
That's really the situation you see on the ground.
Yeah, it's a whole separate, I think, discussion that we could have on that for another hour. But I think in the interest of time, we need to move on. Further questions, obviously, welcome in the break.